Hello, viewers. So today our topic is these factors affecting immunogenicity. So the factors which affect immunogenicity are the first thing is the nature of the protein. So you know that proteins are the best immunogens. And then the conjugates of the proteins with nucleic acids, with lipids, with carbohydrates, they will perform as the best immunogens. So here we see that there's amino acids and then haptens and then lipids and then steroids and then carbohydrates and then simple peptides and proteins and then complex proteins. So here you can see that it is increasing the immunogenicity of the molecules. Means amino acids will be not immunogenic, but haptens will be immunogenic when they are bound with the carrier proteins, and lipids will be more immunogenic. Steroids will be more immunogenic as compared to lipids, and these haptens and carbohydrates will be more immunogenic than steroids, lipids, and haptens, and simple peptide and proteins will be more immunogenic then carbohydrates, steroids, lipids, and haptens, and the complex proteins will be the best immunogens. So here you can see the increasing immunogenicity. That amino acids, from amino acids to haptens, and from haptens to lipids and steroids and carbohydrates and simple peptides and proteins and complex proteins. So the factors which are affecting immunogenicity are the first one is the nature of the compounds. So the best immunogens are the proteins. The second thing is the size of the molecules. If the molecules size is smaller, they are not immunogenic, or maybe they will have very less immunogenicity. But when we increase the size of the molecules, then its immunogenicity will increase. So the first thing is the nature of the molecule. The second thing is the size of the molecules. When the size of molecules will increase, the immunogenicity will increase. And the third thing is the complexity of the molecules. So here you can see that simple peptides and proteins are less immunogenic, while the complex proteins and peptides are more immunogenic. So complexity means, for example, there can be some proteins, for example, collagen, that it is just made up of 3 amino acids, repetition of 3 amino acids, glycine, proline, hydroxyproline, glycine, proline, hydroxyproline. So this collagen will be not immunogenic because it have very homogeneous structure and no complexity in it. Of course, it can have larger size, but since it will have no complexity, no heterogeneity, then it will not be immunogenic. So these complex proteins will have a different sequence of these amino acids, and therefore it will produce different epitopes, and when there will be different antigenic determinants epitopes, then it will be more immunogenic. So, so size of protein is important, but in addition to the size of protein, the degree of heterogeneity is much more important because it is responsible to produce these different epitopes. And when a protein will have more epitopes, then it will be more immunogenic. So first thing is the nature of the molecules, and the second thing is the size of the molecules, and the third thing is the complexity of the molecules. And the fourth thing is the degradability. Means those molecules which are degradable, means the enzymes are there in the living cells which can act on it, and in most of the cases, it's the proteins which are larger in size, which are complex in nature, and which are heterogeneous in nature, and which are degradable because the cells will have these proteases which will act on it and will destroy and degrade those proteins into, will process those proteins into small pieces so that those small pieces will bind with the MHC, and then MHC will bring it to the outside to activate the TH cells and TC cells. The processing of protein is very important because the epitopes of T cells are hydrophobic in nature, and they are in the core of the protein, and they have to be taken out. And for that purpose, the proteins first, they have to be processed and to make the fragments of the proteins with a size of about 10 to 13 amino acids. So they will be activating the T cells, TH cells, and TC cells, because the immune response is not just production of antibodies, it's also production of these TH, activation of TH cells and TC cells. So here we see the degree of immunogenicity, that amino acids or glycines will be not immunogenic. But if they are immunogenic, they will be very less immunogenic. Because there are two things here. One thing is that this molecule, if it's foreign, but it cannot be foreign for our body because our body already have this glycine in it, and immune system knows that it is self. And when it is self, then immune response or immune system will not act against it. So amino acids to haptens, and from haptens to lipids, and from lipids to steroids, and then carbohydrates, simple peptides, proteins, and complex proteins. So, so these carbohydrates and these steroids or lipids, they will be more immunogenic when they will come and in the form of conjugated with the proteins. Then they can work as more immunogenic molecules. But when they will come just by itself and no proteins involved, then they will have very less immunogenicity. So here we can see that these factors governing immunogenicity, in general, the greater the size and complexity of the antigen, the greater the variety of possible epitopes, and the greater the immunogenicity. So all these things are interconnected with each other. So main thing is the epitopes. And epitopes will be more on the proteins because they will have more heterogeneity, and they will have greater size, and they will have more complexity. And when they will have more complexity and larger size, then they will have more epitopes. And epitopes are the sites against which epitopes will be produced. And the size of the epitopes will be just about a page of 6 amino acids. So if we have a protein of 300 amino acids, it can have maybe 30, 40 different epitopes. And against each epitope, since the epitope will have different sequence of amino acids, then there will be a different antibody required for it. So anyway, here we have this, that immunogenicity will increase when the size of protein will increase, and also the complexity of the protein will increase, and also the foreignness of the protein will increase. Because foreign is important, because the proteins we have in our body, simple proteins, we have complex proteins, we have heterogeneous proteins, but they are not immunogenic because they are self. So, and also, for example, if we take example of this bovine serum albumin, so it will be not immunogenic. If we take it out from one cow and inject in the other cow, it will not be immunogenic. But when I take this BSA and inject in the goat, then it will be immunogenic because somehow it will have different sequence from this albumin of goat, since it belongs to the cow, so it will be different. So, but it will not be so much different. It will be more different in case of rabbits. So in case when we inject this BSA in the rabbits, it will be more immunogenic as compared to goats. And when we take this BSA and inject in the rats, then it will be more immunogenic because the evolutionary distance of these rats is more as compared to the goats from cows. So anyway, when the evolutionary distance of the animals will increase, then the proteins will change, and protein will have different amino acid sequence. And when they will have different amino acid sequence, then they will have more degree of foreignness. And when there will be more degree of foreignness, then they will be more immunogenic. So we cut to short, we have these, the factors which affect immunogenicity are the nature of the molecule, the foreignness of the molecule, degree of foreignness, how they are different, how much they are different, and then the size and complexity and degradability of the proteins. Okay, thank you.